need to position myself exactly where I was told. Here? No, here? Is it good? Am I at the right distance? Okay, welcome everyone to Rachel Clifford's thesis defense. Thank you all for coming. It's enough. We are done. Okay, goodbye. Thank you. All right, so thank you all for coming. Our family, also our friends in Lancaster, Pennsylvania. Apparently, we have a crowd watching online. It's like the DNC, basically. Also, I'd like to welcome one of her thesis advisors, actually joining us from nearby Lancaster, from Lafayette College. And yeah, everyone, thanks for coming on a Friday morning. So this is our first thesis defense of the semester. And yeah, it's exciting, right? So I'll give you a few information about Rachel, and then she'll take over, and we'll finish in about 45 minutes for questions, and then we'll disappear in the back room, and we'll come back with her being very happy. That's the plan. That's the plan. So Rachel got her bachelor degree at Lancaster, at the what is it? Franklin and Marshall College. Okay, I have my notes, but I want to try to remember. And in environmental science, right? And then she joined Mossland in 2021. And actually, she joined Dr. Sarah Smith's lab. And she was a GA for her in the classes called Molecular Techniques. That's actually being taught this semester. And after a year or so of investigating molecular-related thesis, she got really interested in biogeochemistry. And since I'm not a biogeochemist, she joined my lab. Because I just got back from a cruise, a deep sea drilling cruise in the Guaymas Basin in the Gulf of California, where we found really weird things, good weird, right? And Rachel got really interested in the biogeochemistry of those marine sediments. So what she did, she came up with different ways to test different hypotheses concerning why those sediments look weird. I'm not going to give you any, I don't want to give up the story yet. But what I want to say is that throughout her experience here at Moss Landing, Rachel has been a GA in molecular techniques, chemical oceanography with Dr. Grant, and in scanning electron microscopy and X-ray diffractometry with me. So she really is one of the examples of how Moss Landing is an interdisciplinary place where she really worked across labs. And her thesis reflects that because the tools and the applications she used to answer those questions are indeed multidisciplinary. The other thing that I want to bring up is that she was a recipient of the Martin Scholarship this year, which is by far, the most important scholarship we have here. John Martin is the founding father of Moss Landing, and she's taking the carbon cycle to a whole different level. That's what her thesis is about. So I think it's going to be really exciting to learn what she did. And finally, she just won a competition on horse ride. So I know there are horse riding fellows here. So that's like the other half of her life, or maybe, I don't know which part of proportion of her life, but that's also very exciting. And the last thing I learned about her, that you play trumpet.
I do play the trumpet.
You never told me. We should play together.
I was late a long time.
Oh, okay. Well, it's still in your CV. Well, so join me in welcoming Rachel's thesis on the analysis of solid phase mineralogy to explore past biogeochemical conditions of the deep biosphere in the Guaymas Basin. Thank you. All right.
All righty, folks. Thanks so much for that introduction, Ivano. Hi, I'm Rachel. And I'm going to be talking to you today about my thesis work studying the deep biosphere in the Guaymas Basin and using the solid phase to reconstruct biogeochemical conditions of the deep biosphere. So first things first, the Lord of the Rings. For all the lay people in the audience, I really hope that you can approach this talk as you would approach watching The Lord of the Rings for the first time. We might get into kind of the lore of biogeochemistry and geochemistry in general, but hopefully I'll bring you back to Earth and back to the main plot line often enough that you'll be able to follow the story and know that by the end, Frodo is going to throw the ring into Mount Doom. So as a general layout of where we're going to go in this talk, we're going to start with my research background and motivation. Then we'll go into my hypothesis development and question development. Then we'll dive into the methodology of how I explore those questions. Then we'll follow up with the research results, and finally, my discussion and conclusions based on those results. So starting, why are marine sediments so important? Why are they worth studying? We like to think of marine sediments as a really important potential source of sinking organic carbon from removing CO2 from the atmosphere and fixing it in marine sediments. That works using the biological carbon pump, where we have phytoplankton growing in the surface waters, fixing CO2 from the atmosphere, and approximately 1% of those phytoplankton die and then settle all the way to the deep ocean into marine sediments, where they could be potentially sequestered and stored for thousands, hundreds of thousands, even millions of years. So that provides a major potential mechanism for how we can sequester a tremendous amount of carbon from the atmosphere into marine sediments, and is why marine sediments are so worth studying. The ultimate fate of that organic carbon, once it's buried in those marine sediments, is something that we're still exploring, particularly in sediments that are disturbed. For example, if you have a tremendous amount of rifting and the intrusion of some really hot magma into those sediments, that can really alter the situation, which is what we're going to go into detail here. So here we have a deep spreading center within marine sediments. This whole light brown area is the marine sediments. The blue is the overlying water. And when we have a rift basin, we have a spreading center where that magma is intruding into those sediments and forming what we call basaltic sills, where that magma is spreading out horizontally, introducing a tremendous amount of heat into the sediments, which very much alters the physical, chemical, and microbial processes that are occurring in those sediments. The organic carbon that settles into those sediments can be buried and remain as organic carbon. It can be stored in the long term as methane hydrates or reprecipitated as carbonates, which again we'll store for the long term. But if that organic carbon remains as organic carbon and is introduced to a tremendous amount of heat from those sill intrusions, that can pyrolyze and sort of cook that organic carbon, making it less available. It can power fluid advection from the sediments and result in the release of that organic carbon in the form of CO2, methane, alkenes, petroleum, and other light hydrocarbons from the sediments, which is what's detailed by those little wiggly lines up there. And so if we want to understand the ultimate fate of organic carbon in marine sediments, how long it's going to stay down there, it's really important to study spreading centers like this. And that was the primary goal of the International Ocean Discovery Program, Expedition 385, to the Guaymas Basin. So Expedition 385 went to the Guaymas Basin on the Joides Resolution, this large vessel here with that really big platform on top of it. And it penetrated the sediments, probed down hundreds of meters below the sediment surface, and we were able to take samples directly from the deep biosphere. On the far bottom right there, you can see some scientists, likely including Ivano potentially, carrying in some of those samples, some of those core samples from the Joides Resolution. So essentially we're poking holes in the seafloor and directly measuring these carbon cycles and the geochemical conditions of the sediments. So the Guaymas Basin is located in the Gulf of California, right in the middle of the Gulf of California there, along a major rift basin and a spreading axis. The Guaymas Basin is characterized by really high productivity in the surface waters and a high sedimentation rate as well, which means lots of organic carbon that is being fixed in the surface waters and then is settling into those sediments. So those sediments are very rich in organic carbon. The sedimentation rate is about 1 meter per 1,000 years. And it's also characterized by rapid seafloor spreading, which means the introduction of those thick, hot, igneous sills into the sediments, introducing lots of heat and changing the balance of the carbon cycle in those sediments. So on the far bottom right there, we have a bathymetric map of the Guaymas Basin, and that long orange, well, maybe red line is a transect. along which there are multiple sites that were studied. That transect is depicted in that upper figure there, which is a seismograph of that whole transect. And basically, the denser the sediment, the more that sound is reflected off. And so those red lines indicate some really dense, intrusive igneous sills. The spreading axis is here in that red arrow. It's also down below where that other red arrow is on the bathymetric map. And the two sites that I'm interested in studying, site 1545 and 1546, are right next to each other, but sit about 55 kilometers away from that spreading axis. And despite being tens of kilometers away from the spreading axis, there's still large igneous sills present there. So that spreading axis has influenced tens of kilometers beyond the axis itself. So site 1545 and 1546 are really valuable test sites. They're only 1.1 kilometers apart from each other, and they have extremely similar sediment profiles. And so they can sort of act as like twin sisters. They're in a very similar place, but the major difference being that site 1546, which is on the right there, in that zoom in of the same seismic profile, has a large intrusive igneous sill about 300 meters below the sediment surface. And if you look at that seismic profile, you can see the intrusion of that sill has caused a significant amount of disturbance at that site. Importantly, some of that disturbance comes in the form of some compaction and dewatering of the sediments by about 30%. You can see that as those lines sort of compress over by site 1546. And the timing of that intrusion is estimated to be between 50,000 and 100,000 years ago. So again, we have these twin sites where site 1545 is largely undisturbed by those intrusive igneous sills, whereas site 1546 is massively disturbed by a large intrusive igneous sill. Going into some of the nomenclature and terminology, and a little bit of background on what I'm going to be talking about. You're going to hear me talking pretty extensively about pore water and the solid phase. When I say pore water, I'm talking about water that is filling the gaps between sediment grains, and it contains ions from seawater and ions produced by microbial metabolism. And that's denoted here on this figure in those like little blue bits. That's the interstitial space between sediment grains. When I'm talking about the solid phase, I'm referring to the sediment grains themselves. They're the solids making up the sediment, and their composition is determined both by the particles that are settling from the overlying water, including those phytoplankton, and the minerals that are precipitated directly from pore water. So we have both in situ and ex situ components to the composition of the solid phase. Diving into a little bit of the background on carbonate chemistry. Calcium carbonate is a solid mineral that forms through the combination of calcium ions and bicarbonate ions. And there's many different forms that carbonate can take. First, we have biogenic carbonate, which is depicted here on the left. This is a coccolithophore. Biogenic carbonates are formed as part of the skeletons of marine organisms. And a coccolithophore is a phytoplankton that grows near the surface that then has settled into the sediments depicted here. Then we have authigenic carbonates. Authigenic means that it formed in situ and is often precipitated directly from pore water. And that's what is depicted over here on the right. These are authigenic dolomite rhombs, those sort of rhombic crystals that you're seeing there. They're fairly large in size, and so not as large as the authigenic carbonates that I'm going to be looking at. I'm mostly going to be looking at authigenic carbonates that take the form of micrite, where micrite is an authigenic carbonate that precipitates and is less than 4 microns in diameter. Calcite is a carbonate mineral composed entirely of calcium and carbonate, and dolomite is a carbonate mineral composed of calcium-magnesium carbonate, where there's an equal 1-to-1 ratio of calcium to magnesium. So that calcium is substituted out by magnesium exactly half the time. Now diving into a little bit of microbial metabolism and its influence on pore water chemistry and how it influences the solid phase as well. So there's a really complex microbial community that lives in marine sediments that utilize a variety of different electron acceptors to power their metabolisms. The two primary metabolisms that I'm going to be talking about today are sulfate reduction, which is that equation that you see on the very top on the far right there, and methanogenesis, which is the equation you see on the very bottom on the far right there. The transition between sulfate reduction and methanogenesis in marine sediments is referred to as the sulfate methane transition zone, or the SMTZ, and you'll hear me refer to the SMTZ throughout. Right at the SMTZ, there's a unique metabolism known as the aerobic oxidation of methane, which couples sulfate reduction with the oxidation of methanogenesis to produce energy. The anaerobic oxidation of methane, or AOM, which is in that middle reaction there, where that arrow is pointing, produces a tremendous amount of bicarbonate, which really influences the alkalinity of the water. Alkalinity is the buffering capacity of pore water, and in this pore water, it's determined by the concentration of CO2, bicarbonate, and carbonate ions. And if you look at the reaction on the left here, which is the carbonate precipitation reaction, if you have a lot more bicarbonate in your pore water, you're going to be favoring the precipitation side of that reaction. So high alkalinity usually favors carbonate precipitation. This led me to develop sort of a model of how pore water and microbial metabolism influences the solid phase. So aerobic respiration and nitrate reduction produce a lot of CO2, which lowers pH and favors the dissolution of carbonates. So on this figure on the right here, we have pore water depicted by all those lines, and then we have the solid phase on the right. In the upper portions of the sediment, we would expect to see carbonate dissolution occurring where nitrate reduction and aerobic respiration are occurring. Deeper down, however, around the SMTZ, where the anaerobic oxidation of methane is occurring, that's associated with really high alkalinity, which you can see in that red alkalinity line, which induces the precipitation of authigenic calcite right around the SMTZ. And dolomite precipitation, which is, that, again, that calcium and magnesium with carbonate crystal lattice structure, is prohibited from precipitation by the presence of sulfate. Sulfate forms a strong ion pair with magnesium, and so inhibits the precipitation of sulfate. So we don't expect to see dolomite precipitation occurring until below the sulfate reduction zone, so usually below the SMTZ. So in summary, we expect to have carbonate dissolution occurring in the surface portions of the sediments, authigenic calcite precipitation occurring around the SMTZ, and dolomite precipitation occurring just below the SMTZ. And this is our model for how we expect pore water to reflect the solid phase. This is the model that we're primarily going to be testing in my project, and the questions that we're going to be addressing address this model directly. The solid phase is accumulatory in nature, and so is more of a time-averaged view of the past. It allows us to reconstruct what has happened in the past over all time, whereas pore water changes relatively quickly. And that's a concept depicted here, where the microbial community alters pore water quickly, and therefore pore water provides a snapshot of current microbial metabolism and the biogeochemical conditions of the sediments today. So current snapshot. Whereas the solid phase accumulates over time, so therefore the solid phase provides a time-averaged view of the past. Using the solid phase as a time-averaged view of the past is a concept I borrowed from the world of conservation paleobiology, and is kind of the crux of what we're going to be testing here, whether or not we can reconstruct the past using the solid phase. So my primary questions and hypotheses. The first question is, can we even use the solid phase to reconstruct biogeochemical conditions of the past in the Guaymas Basin? And we're going to do that by looking at Site 1545. It's our undisturbed control, so there's no large intrusive magmatic sill that's altering the sediments. So I'd expect, with the lack of disturbance at Site 1545, that the solid phase would be fairly consistent with pore water chemistry at that particular site. The second question addresses, how does intrusive magmatism alter the biogeochemical conditions of the Guaymas sediments? The way I'm going to be addressing this question is using Site 1546, whose large magmatic intrusion has caused significant disturbance, and due to that magmatic intrusion, I would expect the solid phase would be inconsistent with pore water chemistry at this site. Now diving into the methods of how exactly I pursued these questions and analyzed the solid phase. I started by using smear slide analysis, which provided petrographic data on micrite and coccolith percent cover. I made individual smear slides for each sample at both sites, 172 slides in total. And I analyzed them using this microscope here on the bottom right, which is a petrographic microscope, and I looked at these slides under polarized, cross-polarized light and parallel polarized light to estimate percent cover. In the middle here, we have an example of what coccoliths look like under polarized light, these little clusters of 4 dots. Altogether, that's what coccoliths look like. That provides an estimate of biogenic carbonate in the sediments. And then on the far right, we have low coccolith percent cover, higher micrite percent cover. And those high bipharengiate grains there, those kind of like rainbowy colors, are indicative of micrite. They extinguish as you rotate the stage. So I used these to estimate how much coccolith percent cover was in the sediments and how much micrite was in the sediments. I performed 5 replicates of percent cover estimates on each slide on all 172 slides. This is what the slides look like. It's a very thin film of sediment that's spread out across the slide that's then examined under the parallel and cross-polarized light. The next major technique that I used was X-ray fluorescence, which provides elemental composition data. X-ray fluorescence works by using an X-ray to knock off an electron from a low energy orbital, one of those inner orbitals. That's the incident X-ray in that bottom sort of figure and diagram. When it knocks off that inner electron from a low energy orbital, an electron from a higher energy orbital falls to fill that void. When that occurs, it releases energy in the form of an X-ray. The wavelength of that X-ray is determined by the amount of energy that's released when that fall occurs. And so the spectrum of X-rays released are unique to each element and is what allows us to determine the elemental composition of the sediments. Our X-ray fluorometer identifies elements from magnesium to uranium, so not the entire periodic table, but a good portion of it and portion concerning the elements I'm interested in. And I used X-ray fluorescence in triplicate on 156 samples that are all contained in those tiny little cups. That large ray gun looking thing is the X-ray fluorometer itself, and the little white cups next to it are the powderized sediment. I dried that sediment in a drying oven, powderized it to be less than 61 microns, and then analyzed it using the XRF. The next method of analysis that I used was X-ray diffractometry to collect mineralogical composition data. Similar to XRF, X-ray diffractometry also uses X-rays to perform its analysis. If you focus on the upper diagram there, we have an incident X-ray coming in to a crystal lattice structure. So those we have the atomic planes there. Those are those black lines with the dots on them indicating the single atoms. If you can imagine that as like a 3-dimensional crystal. The incident X-rays are bouncing off of the crystal planes and being reflected at an angle. And at just the right angle, the peaks and troughs of the reflected waves line up with each other, and that is constructive interference and results in a rapid increase in intensity that we can identify. The angle at which those waves constructively interact is related to the distance between the atomic planes. That's because the atomic planes have a similar distance to the wavelength of X-rays. So we can use Bragg's law, which details the relationship between the angle of constructive interference to the atomic distance between atomic planes, where the wavelength of the incident X-rays equals the distance between the atomic planes times the sine of the angle of constructive interference. So that means we produce these spectrograms that you see on the bottom here. On the y-axis, we have intensity, and on the x-axis, we have 2 theta, which again is that theta is the angle at which constructive interference is occurring. And so we see a peak associated with a particular crystal or a particular mineral. So here, most notably, you can see the quartz peak, which is the peak that I used to align my data, because we had a lot of quartz in all of our samples, so it was really easy to use quartz to align everything. Then we have feldspars. There's a lot of substitution of ions in feldspars, so that's a little bit wider in terms of peak. Importantly for me, we have the calcite and dolomite peaks, halite, which is just table salt, and then pyrite at the far end. XRD analysis was run on a subset of 16 samples, and I determined which samples to utilize based on smear slide and XRF data. I chose some end members that I felt like would represent the mineralogical composition of the sediments fairly well, and that were localized around particular areas of interest. And the final method of analysis that I utilized was scanning electron microscopy and energy dispersive X-ray analysis. So scanning electron microscopy, or SEM imaging, provides high magnification images that you can see on the top right there. That's a zoomed in image of a coccolithophore, similar to what we saw at the beginning. And then EDX provides elemental composition, similar to what the X-ray fluorometer did, except instead of using an X-ray to knock off those inner electrons, we're using a beam of electrons to knock off those electrons. Using EDX and the SEM together allows for the identification of specific grains within a field of view. So we can actually map particular elements across a field of view, which you can see on the bottom right there. All those little red dots are individual points that were analyzed, and the red dots indicate where there was iron present in the sediment. And you can see there's kind of a hot spot of iron around that giant soccer ball looking thing. That's a pyrite framboid. Pyrite is composed of sulfur and iron, so you would expect to see a hot spot of iron right around a pyrite framboid. You can also do point ID on those specific grains to see if the atomic ratio is consistent with what you would expect. So you can do analysis to see if, like, in fact, sulfur and iron are at the ratios you'd expect for pyrite, to confirm that that is, in fact, pyrite. SEM EDX analysis was performed on the same subset of samples that I used on the XRD, so I could kind of cross-reference the two. And to make these samples, I took a very small amount of powder from the samples themselves that had been dried and sieved. I adhered them to an SEM stub using double-sided carbon tape, then used copper tape, which is those little golden rectangles there, to use as a reference for the EDX analysis. And then it was coated in gold using a sputer coater from the 1970s, which increases the conductivity of the sample and prevents electrons from accumulating on the surface. So what were my results? Starting with the pore water chemistry. Pore water chemistry was analyzed on board the ship using an IGPMS, and these are the results from that pore water chemistry analysis. So we have site 1545 on the left and site 1546 on the right. And this is depicting sulfate and methane concentrations with depth being on the y-axis, going from 0 all the way down to 500 meters below the sediment surface. So sulfate is in the blue, methane is in the red, and you can see that sulfate decreases really quickly within the first 50 meters or so at site 1545. And then once sulfate reaches its minimum, methane starts to increase. That's a really clear indication of where the sulfate-methane transition zone is at site 1545, and so it occurs right about 55 meters below the sediment surface. At site 1546, we see the same pattern, but occurring much deeper in the sediments. So at site 1546, we can see that the sulfate-methane transition zone is occurring at about 100 meters below the sediment surface, nearly twice the depth of where it occurs at site 1545. Which is a pretty important point, something unusual that we're going to be exploring as we look at the solid phase. But continuing with pore water chemistry, we're looking at the alkalinity and calcium ion concentration in these sediments. Again, 1545 on the left, 1546 on the right. And we have alkalinity depicted in the red and calcium ion concentration depicted in the blue. Alkalinity, as I mentioned before, is a measure of the bufferability of the pore water, and is strongly controlled by the amount of bicarbonate that is produced as a result of anaerobic oxidation of methane. So we expect to see alkalinity peaking right around the sulfate-methane transition zone where AOM is occurring. And that's exactly what we do see at both sites. We see alkalinity peak right around 50, maybe 60 meters at site 1545, and down around 100 meters at site 1546. A high alkalinity, because it favors the precipitation of carbonates, means that we're going to be drawing calcium ion concentration out of the pore water. So we expect to see a minimum of calcium ion concentration where we have a maximum of alkalinity. And that's what we see at 1545 as well. 1546, it's a little higher than we'd expect, but not terribly out of phase with each other. So in summary, alkalinity peaks and calcium ion concentration drops right around the SMTZ at both sites, just as we expect to see. Going into the solid phase analysis, this is the work that I did using the X-ray fluorometer. So this is the elemental composition with regard to calcium at site 1545 and 1546. The individual dots, those are the actual data points. The dotted line that you're seeing is the moving average, and the ribbon, the shaded area, is the 95% confidence interval. And generally, we see a very similar pattern in calcium weight percent at both site 1545 and 1546. Namely, we see a decrease in calcium through the upper portions, the upper 30 meters of the sediments at both sites. And then we see calcium concentrations start to increase again as we get deeper down. At 1545, that increase occurs at about 70 meters below the sediment surface, and at site 1546, that increase in calcium occurs right around 55 meters below the sediment surface. So calcium weight percent shows the same general pattern at both sites, where we have a decrease at the surface and an increase in calcium at depth. Moving on to smear slide data, we have coccolith percent cover depicted in these two figures on the left, again 1545 on the left and 1546 on the right. And then on the far right, we have micrite percent cover. These are our proxies for biogenic and authigenic sources of carbonate, and coccolith and micrite percent cover allows us to parse out whether or not the carbonates are authigenic or biogenic in nature. Overall, we see coccolith percent cover is decreasing with depth at both sites, whereas micrite percent cover is increasing with depth at both sites. Because these are representing the biogenic and authigenic phases of carbonate, if we add these two together, we should get kind of a proxy for total carbonates in the sediments. It's not a perfect proxy, but it should be a good estimate. And when we add them together, this proxy for carbonates really reflects the same pattern that we observed in the calcium concentrations in the sediments. So similarly, we have carbonate percent cover decreasing in the surface sediments, and then at site 1545, carbonate seems to increase below about 75 meters. And at site 1546, we see carbonates increasing at about 55 meters below the sediment surface. So the sum of micrite and coccolith percent cover mirrors the trends that we observed in calcium weight percent. If I plot these two against each other, where I plot calcium weight percent versus smear slide carbonate percent cover, we have a really strong positive correlation between the two. We have site 1545 depicted in the red and site 1546 depicted in the blue. The lines indicate a significant positive regression, linear regression, and the shaded area, again, is the 95% confidence interval. I also performed a test to determine that the slopes are in fact equivalent between these two sites. This tells us that calcium weight percent is a good proxy for carbonate content in the sediments, and that's a really important finding. These are two completely independent analyses that I performed, and so that confirms that I can use calcium weight percent as that proxy for carbonate content. I then plotted calcium weight percent versus calcite specifically. This calcite data is coming from the X-ray diffractometer, so the 16 end members that I identified to run it using under the XRD to determine mineralogical composition. And I found, again, a significant positive correlation with an R squared that's fairly high, of 0.73. So despite the smaller sample size, we still have a significant positive correlation between calcium weight percent and calcite percent in the sediments. This demonstrates that calcite is a major contributor of calcium to the sediments. So a lot of the calcium that we're seeing in the sediments are coming in the form of calcite specifically. So calcium can be used as a proxy for calcite as well. Moving on to how we determined dolomite in the sediments and how we're estimating dolomite. So all of that was focused on carbonates and calcite specifically. This is more focused on dolomite. When I measured dolomite under the X-ray diffractometer, I only found dolomite in the deepest 2 samples, which tracks with the fact that you would expect dolomite to be occurring below the sulfate-methane transition zone because of the inhibitory effect of sulfate on dolomite precipitation. So there's no dolomite found in the sediments above 60 meters. However, if we look, that's in this figure on the left here, that's the XRD data. If we look on the figure on the right-hand side, that's magnesium weight percent determined from X-ray fluorometry data. We can see that there is a significant amount of magnesium in the surface sediments. So this is telling me that the magnesium that's occurring in the surface sediments is not the result of dolomite, because XRD has determined that there is no dolomite above 60 meters. So because dolomite does not appear to be a source of magnesium above 60 meters, I chose to focus my correlations between micrite and magnesium weight percent in the lower 60 meters of the sediment, because I know that there are other minerals contributing to the magnesium in the surface sediments. So when I did that, I plotted magnesium weight percent from XRF data versus micrite percent cover from the smear slide analyses on the samples below 60 meters. And I found a significant positive correlation, again, at both sites, a stronger correlation at site 1545, but the slope of those 2 lines are parallel, meaning that the relationship between magnesium weight percent and micrite percent cover is equivalent at both sites. So this relationship could indicate that micrite below 60 meters includes dolomite. It includes calcite as well, but it also includes dolomite, and suggests that below 60 meters we could use magnesium weight percent as a way to estimate where dolomite might be starting to precipitate, because we don't have the resolution from the XRD data to determine exactly where dolomite is starting to form. So moving on to the SEM and EDX analysis, this confirms that we do in fact have dolomite present in the sediments because we can visually see it. On the far left, we have 1545 above and 1546 below. On the far left of both of those figures, we have the zoomed in higher resolution SEM images of some dolomite crystals that I identified. I identified them using EDX, which is these maps on the right here with all those colored dots. The red dots are identifying points that contain calcium. The green dots are points that contain magnesium. And anywhere where there's yellow dots is points that contain both magnesium and calcium. So I found points where there was a lot of overlap between magnesium and calcium. And then I did point ID on those particular points. And that resulted in these tables that you see on the far left, where we can get atomic percent of that particular point. And if we look at the atomic percent of magnesium and calcium in both of these points, we can see that magnesium and calcium are at an equal atomic ratio, which is what you'd expect to see in dolomite, which has a 1-to-1 ratio of calcium to magnesium, which confirms that these crystals are in fact dolomite. And this upper crystal that I took a picture of at site 1545 demonstrates those same ROMs that we saw, and you would expect to see it in dolomite, micritic dolomite, which is pretty exciting. So if we take all of that data that I just spewed at you and compile it into a single figure, that's what we get here. So what exactly is the carbonate composition of these sediments? This is kind of a complicated figure, so I'm going to go through it very slowly. Once again, we have site 1545 on the left, 1546 on the right, and depth on the y-axis, going down to about 120 meters below the sediment surface. The circular points that you're looking at are calcium determined from XRF analysis, and the blue line is the moving average of calcium weight percent from XRF analysis, again with the shaded area being the 95% confidence interval. So that's those circular points. The color of the circular points is associated with the relative contribution of coccoliths versus micrite. So the more yellow the points are, the higher the proportion of coccoliths versus micrite, and the more blue the points are, they almost look black, the higher the proportion of micrite relative to coccoliths. So up near the surface there, you can see there's lots of yellow points. That indicates a high biogenic carbonate source of carbonate in the sediments versus at depth we can see that there's more of an authigenic carbonate source into the sediments. And then the orange diamonds there are indicating magnesium weight percent below 60 meters, which is our proxy, our estimate of where dolomite is forming in sediments. So, and we have our dotted line, of course, denoting the sulfate methane transition zone at both sites. So at site 1545, well, similar at both sites actually, we have biogenic carbonate dissolution indicated in the upper 30 meters by the decrease in calcium weight percent and the transition of those dots from yellow to more green in color, indicating that coccoliths are decreasing in their abundance in the sediments. At depth, at both sites, we see calcium weight percent increasing again, and we see the increase in calcium weight percent being dominated mostly by sediments that contain lots of micrite, meaning that authigenic carbonates are the contributing factor to the increase in magnesium and calcium weight percent at depth. And we also see magnesium weight percent increasing at depth below 60 meters, indicating dolomite precipitation. So what this means at site 1545 is that we have biogenic carbonate dissolution in the upper 30 meters, and then at about 70 meters below the sediment surface, we have authigenic carbonate precipitation occurring, and authigenic calcite specifically. And we also have evidence that authigenic dolomite precipitation is occurring at about 70 meters below the sediment surface as well. At site 1546, we have, again, biogenic carbonate dissolution occurring in the upper 30 meters of the sediments, and authigenic calcite precipitation occurring at about 55 meters below the sediment surface, and dolomite precipitation seems to occur at about 65 meters below the sediment surface. If we look at the relationship between where we see authigenic carbonates starting to form in relation to the depth of the SMTZ, we can see the SMTZ is associated with the presence of authigenic carbonates at 1545, whereas at 1546, authigenic carbonates are occurring well above the sulfate methane transition zone. And that's particularly surprising to see magnesium increasing at that point, indicating dolomite precipitation, because we know that sulfate inhibits the precipitation of dolomite in these sediments, and so we wouldn't expect to see dolomite forming in the upper portions of the sediments above the sulfate methane transition zone. So then how is solid phase composition of the sediments relating to the pore water chemistry that we collected earlier? So this is site 1545. We have the solid phase composition depicted here on the left. Again, this is our understanding of what the time averaged past looks like. And then we have the pore water chemistry, which indicates today. And we see, we'd expect to see, based on the model that I proposed earlier, that authigenic carbonate precipitation should be occurring right around where alkalinity peaks and where calcium ion concentration decreases. And that is pretty much what we see. It's maybe slightly deeper, but not tremendously. It's definitely not out of the realm of possibility, where we see authigenic carbonate, calcite, and dolomite precipitating at site 1545. This is really exciting because the alignment between the solid phase and the pore water here at site 1545 demonstrates that the model that I suggested at the beginning is correct at this particular site. And at this undisturbed site, we would expect to see the solid phase carbonates reflect the patterns that we observe in pore water. So the time average past and the conditions of today are reflective of one another when there isn't a significant amount of disturbance in the sediments. Moving on to where there is a significant amount of disturbance in the sediments, at site 1546, we have a very different story. So at site 1546, authigenic carbonates appear to be forming right around 60 meters below the sediment surface. Calcite occurring at about 55 meters, dolomite occurring at about 65 meters below the sediment surface, well above the sulfate methane transition zone, and well above where alkalinity peaks, which doesn't peak until about 90 meters below the sediment surface, and calcium ion concentration reaches its minimum around 90 meters below the sediment surface. That is around where we would expect to start seeing authigenic carbonates precipitating. And as I mentioned before, we have magnesium ion concentration, or magnesium concentration increasing in the sediments well above this SMTZ, well within the sulfate respiration zone, the sulfate reduction zone. And that's unlikely because sulfate has that inhibitory effect on dolomite precipitation. So this is fairly exciting because it demonstrates that at site 1546, the conditions are deeply perturbed. So the geochemical conditions have been perturbed by the intrusive igneous sill in a major way, and gives us an avenue through which we can explore how the geochemistry at site 1546 has been impacted by the intrusive sill. In order to understand how the sill has been impacting the sediments at site 1546, we need to reconstruct what these geochemical conditions looked like in the past using the solid phase. And that's what we're going to be doing here. So first we have this red arrow indicating where the SMTZ is indicated in the past based on where authigenic carbonates start to accumulate. That's the red arrow. It's important to remember that the sill intruded approximately 50,000 years ago, and with a sedimentation rate of 1 meter per 1,000 years, that means approximately 50 meters of sediment have accumulated since that sill intruded. And so that places the depth of the sediment surface right about here at this red or orange squiggly line. It's also important to remember that when the sill intruded, it compacted the sediments and dewatered them. And so if we correct for that 30% sediment compaction and the additional 50 meters of sediment accumulation on top of the sediments, that means we have the surface of the sediments up here at 0 in the past, pre-intrusion, these conditions on the far right here. And then we have the sulfate methane transition zone when you correct for that compaction of the sediments occurring at about 40 meters below the sediment surface, which means that if the SMTZ is indicated to be about 40 meters below the sediment surface pre-intrusion, that was a change of 60 meters between the SMTZ before the intrusion occurred and the SMTZ today. So this demonstrates that the sulfate methane transition zone became significantly deeper after the intrusion occurred. Alrighty, so what are our major conclusions that we can draw from all of these results? First, the major conclusion is that I was able to use independent analytical techniques for the first time to characterize the microcarbonates in these sediments. So using disparate techniques, smear slide analysis, XRF, XRD, and SEM, EDX, and these significant positive correlations confirm that I was able to use all of those to determine the mineralogical composition and the character of the microcarbonates in these sediments. And I was also able to disentangle the authigenic and biogenic components of those microcarbonates using the smear slide coccolith and micrite percent cover. The other major conclusions I have with regard to the questions that I initially presented, the first being, can we use the solid phase to reconstruct the biogeochemical conditions of the past? In the Guaymas Basin, essentially, yes. Site 1545 demonstrates that the distribution of carbonates in the solid phase is consistent with pore water geochemistry in the Guaymas Basin, which confirms that we can do paleobiogeochemistry. We can reconstruct the conditions of the past. And at sites where there is minimal disturbance, pore water and the solid phase are in agreement with each other. Our second question, how has intrusive magmatism altered the biogeochemical conditions in the Guaymas Basin? That relates to what we learned at site 1546, which demonstrated that authigenic carbonate and dolomite distribution at site 1546 suggests the SMTZ became much deeper after the sill intruded. That's demonstrated in this figure, where you can see that the SMTZ decreased or increased in depth post-intrusion. And so what exactly are the consequences of this research? One major consequence is that heat introduced from the sill intrusion resulted in the deepening of the SMTZ, and that leads to the logical question of why. We know that organic carbon content in the sediments is a major control on the depth of the SMTZ and the depth of the redox cascade in general, where if you have less organic carbon available, there's less competition for those electron acceptors, so the SMTZ tends to be deeper. So this could indicate a reduction in the amount of organic carbon available to those microbial communities in the sediments, and the subsequent deepening of the SMTZ as a result of the intrusion. So the intrusion produced all this heat, kind of cooked those bits of organic carbon, made them less available, less refractory for the microbes to be able to utilize. And second, a major consequence is we've been able to study a diverse set of, if we can study a diverse set of undisturbed regions of the seafloor, we can kind of confirm that what we observed at Site 1545 is applicable throughout global oceans as long as the sediment isn't disturbed. So we could potentially apply this concept and method of paleobiogeochemistry to other seafloor systems if we study other undisturbed portions of the seafloor. And that would provide us with an avenue of exploring disturbance regimes in those sediments as well, exploring places where there is a lack of alignment between pore water and the solid phase, indicating changes between past and present, and would allow us an avenue to explore why those changes may have occurred. So now, time for acknowledgements. First, I want to thank Ivano, my advisor. He's been instrumental in teaching me and allowing me to use all of his fancy instruments, and for countless multi-hour meetings where we try to figure out what the heck is going on. Maxime Grand for being an incredible professor and advisor and mentor as well. John for discussing the details of coccolithophores with me and coccolith dissolution. I'd like to thank the IODP, the International Ocean Discovery Program, for allowing me access to these samples. And of course, Moss Landing Marine Labs, through whom I wouldn't be getting my master's degree, and also through whom I've been able to utilize those fancy instruments. I want to thank all of the members of the IODP 385 crews and expedition, but I particularly want to thank Dee Dee, Nicolette, and Andreas, all of whom I got to speak with directly, who shared data with me and insights that they learned from the crews and on board the ship. I'd like to thank the Moss Landing Marine Labs GeoOcean Lab, Melina, Heidi, Marcel, and Vicky, and the new members as well. Welcome, I haven't met you yet. And just for being wonderful supports, for sharing your knowledge and educating me as well. The Moss Landing Marine Lab staff, who have gotten me out of countless jams when I do something deeply stupid, like locking myself out many times. Tara in particular, your lack of judgment when I do something dumb is huge. So thank you for that. And of course to the Moss Landing Marine Labs faculty, aside from, of course, Max and Ivano, who I already thanked, who have all educated me, provided a lot of mentorship and support and kindness. We have all of these lovely people. This is my mom, dad, and my brother, most notably, who provided me with like endless support and endless love. My friends, the dweebs, the wizards, the Moss Landing Marine Labs friends I have here, all of the members from Carmel River Ranch who are here in force as well, my extended family, all things new, the mentors I've had at other institutions, including Franklin and Marshall, the MBL, and USC, all my critters, all my animals, and of course the Monterey Bay Ecotours staff who is here as well. These are just some photos of everybody, well, not even everybody, but anyone I've interacted with in like the last 3 years has provided a lot of support, and I'm really grateful to every single one of you for being there for me and encouraging me along the way, so thank you.
Okay, we have a few minutes for questions. I'll pass around the microphone. Questions for Rachel? Oh, those are the tough ones. Excuse me.
That was amazing.
Hello, Dad.
So I have two short questions, if that's okay.
Okay.
Site 1545 and 1546, you used 1545 as your control.
Mm-hmm.
Was that known when those samples were taken?
Yeah, we knew based on the, if we go all the way back to like the seismic profiles. We knew that site 1545 had that large intrusive igneous sill, and 1546 did not have the intrusive sill, and 1545 did. So we knew that these would be good test sites
Yep.
when this seismic profile was taken.
And then the second question is, the SMTZ decreases in 1546.
Yeah.
And is that because of the intrusive sills?
That's what
I guess my question is, why does the
SMTZ decrease? So that was one of an area of like further research that we need to explore. But my thinking is that it's a reduction in the amount of available organic carbon as a result of all the heat that's introduced from that intrusive igneous sill. So essentially that sill cooked and pyrolyzed the sediments, and the organic carbon in the sediments made it less available for the microbes to utilize. That lack of competition between the microbes for electron acceptors results in a deepening of the sulfate methane transition zone. There's a little bit more evidence that kind of supports that as well. So, for example, in the upper 30 meters here, it's pretty consistent between both sites where we see a decrease in biogenic carbonates in the upper 30 meters, and that's consistent at both sites. Those upper 30 meters were accumulated after the sill intruded, and so should have consistent content of organic carbon at both sites. And so the fact that that's consistent between both sites suggests that the intrusion of the sill resulted in a decrease in the amount of organic carbon available. And also, the alkalinity at site 1545 is much higher. The maximum it reaches is about 60 millimolar, whereas at site 1546 it maxes out at about 40 millimolar, which indicates there's less microbial activity at site 1546, which would further confirm potentially less organic carbon content in the sediments and lack of competition, or less competition for electron acceptors and a deeper SMTZ. Yeah.
More questions for Rachel? Please.
Okay. You might have said this in the beginning, but what caused the intrusion in the first place, or the displacement?
Yeah, so I probably don't need to go all the way back, but basically the Guaymas Basin, there's a whole network of fault lines there. We have, it's in the Gulf of California, so you have Baja California separating from mainland Mexico, and it's a spreading axis. So the two continental plates are separated, or the two plates, tectonic plates, are separating from each other, and that allows magma an avenue to intrude into the sediments. And those sills are magma that's intruded into the sediments and then introduced all this heat, but then cooled. So the sills are now cool, but at the time when they were formed, they were really hot. Yeah.
The performance dance separation. I don't know if there's anyone from that class. Remember the dance? No. Questions for Rachel? We have another minute or so.
Great talk. One of your conclusions is that now we can do paleo biogeochemistry. So what do you think are some of the important questions you can address with that approach now that you've established that?
That's a great question. I think it would be really important to examine more about the geochemical cycle and the carbon cycle contained within marine sediments and like the long-term fate of carbon. So if we can look at other portions of the ocean and establish that disturbance seems to alter the conditions. Like at 1546, the deepening of the SMTZ means that carbonates aren't precipitating until much deeper. And carbonates are a great way to store that organic carbon for the long term. So in terms of carbon sequestration, in a place that's experiencing a significant amount of rifting and magmatic intrusion into the sediments, I would think that that would be a bad place to focus our efforts of sinking organic carbon, whether we're sinking kelp or phytoplankton or whatever we're trying to do, and maybe focusing those efforts in other undisturbed portions of the world's oceans, places where carbonates seem to accumulate and form closer to the sediment surface. I don't know if that answers your question well.
We have questions from afar. We have questions from afar. Hi, Rachel. absolutely amazing talk. You should be so proud.
Okay. Moving on.
Would you be able to?
Thank you, but
There's more. Would you be able to do some of this work in deeper water? Would you need to consider the carbonate compensation depth at all?
oh, that's a good question. The carbonate compensation depth, I don't think we would need to consider as much because this is kind of a different world we're looking at within marine sediments. So alkalinity changes within marine sediments pretty quickly. The carbonate compensation depth is basically where you're too deep for carbonates to settle. By the time they settle close to the sediments, they've dissolved, essentially, which means that the biogenic component of carbonate in the sediments would be non-existent, but the precipitation of authigenic carbonates should still occur because you still have those microbial metabolisms increasing the alkalinity of the sediments and favoring the precipitation of authigenic carbonates.
Yeah, it was a
And
really
then
good question.
what was the
so
first part of the question?
Oh, that was it.
Okay.
That was another question. How long did the heat last after the intrusion?
Oh, I'm blanking, but I know it's cold now.
It's very cold. We drilled it, so.
Yeah, it's solid basalt at this point.
Yeah, so not too long.
That
right?
is a good question.
A
but
few
yeah, I
thousand
don't know.
years,
Okay.
possibly. So those
Okay.
things get cold very quick. Yeah. All right. Well, thank you for the question. Thank you for coming. Let's congratulate Rachel. And,
I need to open the Zoom link so John
yeah,
can
so
join.
the committee will